Good day, everyone. So, today we will be talking about the chapter 3, which is the culture systems and methods for fishes and crustaceans. So, this discussion focuses on understanding different culture systems, comparing methods, and identifying the most applicable and economically feasible methods depending on environmental and production conditions. So, this report is prepared by the Group 1, which is composed of Jon Michael Faulve, Ethel Joy Labrador, Surainie Mamasabang, Luz Margarette Raganas, Kristhyn Sabanal, and then Maxinne Sumngat. The main objectives of these presentations are to first name and describe each of the culture systems and method of culture, to compare one culture method to the others, and lastly is to identify the best method of culture applicable or adaptable to certain conditions and or in terms of applicability and economic considerations. So here we have the outline of topics for the chapter 3, which includes the types of culture systems relative to confinement, methods of culture relative to species combination, the methods of culture relative to levels of production, the methods of culture relative to natural food, the methods of culture relative to sex of fish, other methods under finfishes, and lastly is the culture methods for mollusks and other invertebrates. So let us first discuss the first major topic of the chapter 3, which is the types of culture systems relative to confinement. Um, Culture systems relative to confinement refers to how aquatic organisms are physically contained during culture. So this includes yung pan culture, cage or pen culture, raceway culture, and the raft culture. So each of these system differs in terms of water control, management intensity, and species sustainability. So for pond culture, it is one of the most widely used aquaculture systems. It is commonly used for species such as for milkfish, shrimp, sea bass, siganid, tilapia, and others. So this system allows partial environmental control and is suitable for both extensive and intensive production. Next, we have the cage or pen culture. So this is usually used in natural water bodies such as the lakes, rivers, and coastal areas like this one in the picture. So this system are suitable for species like grouper, snapper, sea bass, and tilapia. And we also have the milkfish, which is included also in the pond culture, but it's also included in the cage or pen culture and the raceway culture. Now, for the third, we have the raceway culture. So, raceway systems are flow through systems where water continuously flows through culture units. This allows high oxygen availability and waste removal, making it ideal for intensive production and fast growing species. So, in the next slide, I will be showing a picture of what a raceway culture looks like. Last, we have the raft culture, which is for oysters and mussels. So, this is usually a method that maximizes water column space and provides natural feeding opportunities through suspended plankton. So here we have the example of a raceway culture. As you can see, raceway culture or raceway system has this flow-through systems na continuously nag-flow yung water. So, this is very ideal for intensive production. While on the other picture, we have the raft column culture. So, ang raft culture originated actually in Japan, and this technique is used for 3D farming in deep sheltered waters, allowing rapid growth and high yields due to abundant food access. So, usually ginagamit dito is yung bamboo, nag-float na parang rectangular siya. So, yan yung ginagamit sa raft culture. So for the next topic, we have the first methods of culture relative to species combination. So when classified based on species combination, the culture methods include monoculture or the single species, polyculture, and integrated farming. So this method differ based on biodiversity level and the resource utilization efficiency. So first, we have the monoculture or the single species, either saline tilapia, milkfish, siganid, shrimp, prawn, or any single species. So it basically involves only one species in a culture system, which allows easier management, feeding, and harvesting, but may increase disease risk and reduce ecological balance. And next, we have the polyculture or the two or more species. So an example here is a shrimp and milkfish, where the shrimp is the main crop and milkfish is a janitor. So it is usually na species are selected based on complementary feeding habits. So this improves resource utilization and increases total production. So last, we have the integrated farming, um, where it combines aquaculture with livestock or crop production. Waste from one component becomes input for another, improving sustainability, reducing waste, and increasing overall farm productivity. So I have put here an image that shows um, how polyculture works. So yung sa taas, yung A, polyculture system based on ecological process facilitation, shows how different organisms in the pond help each other naturally. So, the main idea here is yung different species, they use different food sources. So, it allows nutrient recycling and better productivity. So, how does it really work? So, yung phytoplankton, will be eaten by the zooplankton and some fish, and the zooplankton will be eaten by omnivorous fish. The aquatic plants will absorb nutrients and provide food or habitat. Then the fish feces and plant residues will become nutrients in the sediment. And then the sediment organisms like the endofauna will help break down wastes, and the bottom feeding fish will steer the sediment and will recycle nutrients. So that is how the polyculture based on ecological process actually works. So the benefit of this is it will improve the natural food production. It reduces waste accumulation. It improves water quality, and it increases overall fish production. So here is a picture of how the integrated farming looks like in a diagram or in a model farm park concept. So, integrated farming, the waste from fish or animals is reused as fertilizer or feed for plants and other farm components. The plants and crops can be used as feed for fish or animals. It produces multiple food sources such as the fish, meat, and vegetables from one farm system. So, it also, syempre, it will improve the resource efficiency and the farm sustainability. So this image shows a small integrated farming system. So basically, how different farm animals, plants, and people can be connected so that waste from one becomes a resource for another. It is about recycling nutrients and reducing waste in agriculture. So instead of raising animals and crops separately, everything is linked in integrated farming. So yung animals, we have here the buffalo, so it will produce manure and byproducts, and this will be used as a fertilizer, as a feed or a fuel. And then the plants grow using recycled nutrients. Ito. Tapos, plants and some animals will become food for humans and other livestock. The buffaloes will eat the litter and feed about 60% maximum in the ration. It then the buffalo will produce excreta, and this excreta will be used as fertilizer or fuel. And then the blood protein, ito blood protein or sludge as protein feed, can be used as a feed supplement. And then the fish, we have here a 1,000 fish. It will use wastewater and byproducts from other farm parts, and then it will produce fish or prawn meat and provide food for people. Um, Next is the plants. So the plants will use animal manure as a fertilizer, and then nutrient-rich water from fish ponds, this wastewater and byproducts, and the plants will then provide or produce grains and vegetables, which will serve as a food for people and a feed for animal. So next we have the broilers or the chicken. So this chicken will eat grains and plant materials, and pag nakain nila yun, maging source of food sila for people. So, as you can see here, the center of the diagram is the human. So, humans will consume chicken meat, fish, prawns, vegetables, and grains. Yung mga araw na nakaturo sa kanila. Then, we'll use also fuel from animal waste. So, next topic is the methods of culture relative to levels of production. So for this topic, I have decided to put it into a tabular form so we can compare and contrast the differences between the methods of culture, kung ano yung extensive, what is the difference of extensive to intensive. So we can obviously look at this table and easily understand or easily compare and contrast the different methods of culture relative to levels of production. So let us first start with the extensive. So ang extensive, its main food source is only natural food. So walang any additional supplemental feed or formulated feed. Then its stocking density is low, which is usually approximately 2,000 to 3,000 hectares allocated for bangus, or 5,000 to 30,000 hectares allocated for tilapia. Then the water depth is usually 50 centimeter or less. Then the investment level, since maliit lang naman yung stocking density mo, so talagang minimal lang siya. Then the yield level is also low. Usually is 700 to 1,000 kilogram hectares per crop. Then for the next method of culture, we have modified extensive. So dito sa modified extensive, para siyang extensive pa rin siya, pero we add supplemental feed, 'di ba? Dito sa main food source. So, yung main food source niya is natural food, and then may additional na supplemental feed. Then mas malaki ng konti yung stocking density niya. Approximately 4,000 to 6,000 hectares allocated for bangus, and then ang water depth niya is 75 to 80 centimeter, and investment level is usually slightly higher, and the yield level is moderate. Then for the semi-intensive, it is a combination of um, an extensive pero mas improved siya, and mas matataas yung mga stocking density and the yield level. So the main food source is natural food plus protein-rich supplemental feed. And then it has a higher stocking density in comparison with the extensive and modified extensive, which is approximately 8,000 to 12,000 hectares allocated for bangus, or 40,000 to 60,000 hectares allocated for tilapia. And it also has deeper water depth, greater than or equal to 100 centimeter. Then investment level is higher, and higher yield level. Then the intensive, where the main food source is complete formulated feed. The stocking density is very high, 15,000 hectares per bangus, hectares allocated for bangus, and then 70,000 to 100,000 hectares allocated for tilapia. Deeper water depth, greater than or equal to 120 centimeter. The investment level is talagang very high siya, and very high yield level. So that is for the methods of culture relative to levels of production. So next we have the methods of culture relative to natural food. Some culture methods depend on natural food availability, such as the plankton method, the love method, and the lumot method. So first we have the plankton method. So I think we are all familiar kung ano yung itsura ng plankton. Um, we watched the SpongeBob, and here is a picture of Sheldon J. Plankton, which is a very famous uh, cartoon character in SpongeBob. So, plankton method. where it is where the fish are reared with plankton as the principal food. So fish may be given supplemental artificial feed to support the growth. Um, Plankton are suspended microscopic plants and animals growing within the water column, as most especially in the upper or surface layer. The plant or the phyto component of the plankton include the green algae, which includes the Senadesmus, the Chlorella, Pandorina, etc. The blue-green algae, which includes Microcystes, Anabaena, etc. Diatoms includes Nitsia, Navicola, Synedra, and etc. The euglenoids, which include Euglena, Phacus, and Trachelomonas. And the dinoflagellates, Serratium, and Gymnodinium. While the animal or zoo components include the Moinia, Daphnia, copepods, and other tiny animals, including the eggs and larvae of the fish, crustaceans, and mollusks. So next we have the lab method. This is where fish are reared with lab as the main food. So similar to the plankton method, we also add supplemental feed to support the growth of the animal. So it is very okay to use additional feed and not only the lab method. So what is lab? So lab or off-watch is an association of phytoplanktons, zooplanktons, filamentous algae, and worms growing at the bottom surface of pond. They are called benthic algae since they grow at the bottom part. The plant components are the blue-green algae, Lingbia, Formedium, etc. Diatoms, the Nitria, Navicula, Cynedra, and etc. The animal components include the Moina daphnia, eggs and larvae of finfishes, crustaceans, mollusks, and insects. So, ang lablab usually kay natural food na yan siya, especially sa bangus and shrimp. So, ang lablab kay para complete mixture na yan siya, hindi lang siya algae, meron siyang plankton, algae, and other organic material. Tapos, usually, ang itsura niya kay brownish, greenish, or yellowish on pan soil. It grows best in dry season, hard pan bottoms, and where the salinity of the water is around 25 to 35 ppt. Next, we have the lumot method. The lumot method is where the fish feed on filamentous grass green algae or lumot as their principal food. So, pwede rin maggamit dito ng supplemental feed like the previous two methods of culture. So, ang lumot kay English niya is moss. So, these are filamentous green grass algae that start to grow at the bottom surface of pond and continue to grow up to the water surface. So, ang lumot kay serve din siya na natural food for bangus and shrimp plus organisms living on lumot are also eaten by the fish too. So it usually grows during rainy season, soft pond bottoms, lower salinity water and water depth with approximately 40 to 60 centimeter. So, ang lumot is harder to digest for young milkfish. So, sometimes, it is dried or decomposed first before ipakain sa mga young milkfish. So here we have the three kinds of lumot. We have the Catamorphalium or lumot GC, unbranched filamentous grass green alga, the Cadophora, which is a branched filamentous grass green alga, the Enteromorpha intestinalis or tubulosa, or also known as bitukang manok. It is an alga which looks like a pancit when it is still young and which looks like an intestine of a fowl when it is in its adult stage.
Now let's focus on the methods of culture relative to the sex of the fish. These methods are specifically designed to promote rapid growth and produce larger sized fish within a shorter culture period. The main goal of these approaches is to control reproduction, reduce overcrowding, and improve overall growth performance. This is especially important in tilapia culture, where uncontrolled breeding can lead to competition for food and space, ultimately slowing down growth and reducing production efficiency. Moving on to the monosex culture, this method involves rearing only one sex of fish, usually males, to prevent excessive reproduction and competition in the culture system. This approach is widely used in tilapia culture, especially saline tilapia, because male tilapia grow faster and reach larger sizes compared to females. By eliminating mixed sex populations, farmers can achieve more uniform growth, better feed efficiency, and higher overall production. To achieve monosex culture, one common approach is manual sexing and hybridization. Manual sexing involves physical identifying and separating male tilapia from females, with only males selected for grow out because of their superior growth rate. Hybridization, on the other hand, involves crossing different species within the same genus to produce fast-growing and predominantly male offspring. Common hybrid crosses can produce about 95 to 100% males, such as crossing male Oreochromis aureus, Mosambicus, or Hornorum with female Oreochromis niloticus. These methods help improve production efficiency while controlling unwanted reproduction. Another widely used technique is the sex reversal method. This method converts genetic female tilapia into functional males during the early fry stage, specifically when the fry are 11 millimeters or smaller and sexual differentiation has not yet occurred. The fry are fed with feeds mixed with the androgen hormone 17-alpha-methyltestosterone for about 21 to 25 days, following a decreasing feeding rate schedule. As a result, a high percentage of male fish is produced, which is ideal for grow-out culture due to the faster growth and better size uniformity. Next is genetic manipulation of sex, specifically the YY male technology. This method produces all male tilapia populations without the need of continuous hormone use. It involves breeding genetically male fish that are capable of producing exclusively male offspring. Although this method is very effective, it is often considered a high-risk technique because of its technical complexity, strict biosecurity requirements, and the need for advanced expertise in genetic management. Lastly, we have the triploidy method. This method produces sterile fish by suppressing normal gonadal development through cold shock or heat shock treatments applied to fertilized eggs before cleavage occurs. Because triploid fish are sterile, sexual maturity is delayed, allowing more energy to be redirected towards growth rather than reproduction. As a result, these fish tend to grow faster and larger, making them suitable for grow-out culture and commercial production. Now let's focus on the other culture methods under Finfishes. These methods are designed to optimize fish growth, maximize pond utilization, and improve overall production efficiency. In milkfish culture, two commonly used systems are the traditional straight run method and the modular method. These two approaches mainly differ in terms of pond layout, fish movement, and the intensity of production. Starting with the traditional straight run method, this culture system involves stocking milkfish fingerlings directly into the grow out or rearing pond. Once stocked, the fish remain in the same pond from the beginning until harvest. Since the fish are not transferred to other pond compartments, this method is considered simple, less labor-intensive, and easier to manage, especially for small-scale or traditional fish farmers. In terms of its characteristics, the straight-run method involves continuous rearing of fish in a single grow-out pond, allowing about 3 to 4 croppings per year. This method is commonly associated with traditional or conventional pond layouts and depends largely on natural pond productivity. There are minimal structural modifications involved, making it cost-effective but relatively limited in production capacity. On the other hand, the modular method of finfish culture is a more intensive and organized system. In this method, milkfish are grown by passing through three progressively larger, grown-out pond compartments. Fish are initially stocked in the smallest pond and are transferred every 15 to 30 days as they grow, until they finally reach marketable size in the largest compartment. The pond structure in the modular system consists of three grow-out compartments known as production process stages, or PPS 1, 2, and 3. These ponds follow a 1:2:4 area ratio, where the largest portion of the pond system is allocated to PPS 3. This design supports a higher fish biomass during the final growth stage and allows continuous and efficient growth as the fish increases in size. Lastly, the modular method offers several advantages over the straight-run system. It allows continuous culture with 6 to 8 croppings per year while reducing pressure on natural food resources. With a complete production cycle of approximately 45 days, this method can yield more than 2 tons annually. Because of its higher productivity and better resource utilization, the modular method is considered more efficient than the traditional straight-run method. Now let's focus on culture methods for mollusks and other invertebrates. These culture methods are designed to improve spat collection, growth, and survival under natural or semi-controlled aquatic conditions. They make use of different structures such as bamboo poles, shells, trays, rafts, and natural substrates to provide surfaces where invertebrates can attach and grow. The choice of culture method depends on environmental conditions, available resources, and production goals, making these methods both sustainable and economical for invertebrate farming. The hanging method is one of the most common techniques used in oyster culture. In this method, cultures are suspended in the water column to allow oyster larvae or spats to attach and grow. Materials such as rubber strips from old tires, plastic ropes with threaded shells, or galvanized iron wires with shells spaced apart are used. These couches are hung on horizontal bamboo poles supported by X-designed bamboo posts, allowing good water circulation and promoting efficient spat settlement. The ring method is a variation of the hanging method and also uses rubber strips taken from old tires. Instead of being suspended with shells, the rubber strips are tied directly onto horizontal bamboo poles and act as spat collectors. Oyster larvae naturally attach to the rubber surface as they settle, making this method simple, low cost, and effective for collecting spats. The stake method uses split or whole bamboo trunks that are driven vertically into shallow, muddy, or soft bottom areas that remain at least 1 meter deep during the lowest tide. Empty oyster shells or tin cans are attached to the stakes at regular intervals to provide more attachment space. The stakes are placed with enough distance to allow oysters to grow naturally on the structures. The tray method uses rectangular trays usually made from bamboo slats, wood, marine plywood, or plastic materials. These trays can be supported by posts pressed into soft mud, tied to fixed poles, or suspended from rafts. This method is mainly used to culture undersized oysters that are separated from harvested stocks and is rarely used as the primary culture method. The raft method makes use of floating structures constructed from bamboo poles or lumber supported by floaters such as sealed gasoline drums or styrofoam. Oyster gulches, similar to those used in the hanging method, are suspended from the raft at regular intervals. Anchors are added to keep the raft stable depending on the conditions of the culture site. The broadcast method is a low-cost oyster culture technique that requires very little capital investment and labor except during harvesting. This method does not require major structures, although boundary markers such as corner posts may be used. Oyster growth mainly depends on natural environmental conditions, making this method suitable for small-scale or traditional farming. To continue, this method is best suited for areas with firm, sandy or rocky bottoms. Materials such as old oyster shells, stones, rocks, iron pieces, or adobe blocks are scattered on the sea or river floor to serve as anchorage. Free-swimming oyster larvae settle on these surfaces as they grow heavier and are allowed to grow naturally until they reach marketable size for harvesting.
So let us proceed to the chapter 4, which is the preparation of the culture units or the ponds. The objectives of this topic is at the end of the chapter, the student should be able to state or list in a sequential order all the main activities and sub activities prior to farming of fish or shrimp in fish ponds as well as in fish cages and fish pens. Second is to cite and explicate the purposes and significance of each sub activity. Third and last one is to enumerate the required production inputs and determine the needed amount of these inputs, such as the lime, fertilizer, fish seeds, feeds, labor, etc., with the use of known formulas. So, uh, heads up na meron tayong mga formulas na i-discuss later sa topic na ito. So, the outline of topics for the chapter 4, we have the draining and drying, tilling or cultivating the pond bottom, leveling the pond bottom, soil sampling, soil conditioning, then fertilization and fertilizers. First, we have the draining and drying. So, we have to dry the pond for about 3 to 5 days until the bottom soil hardens and cracks, like what is on the picture. So, the purpose of drying is to eradicate pests, predators, and competitors, to eliminate obnoxious gases such as the hydrogen sulfide resulting from organic matter decomposition, to hasten the mineralization or decomposition of deposited organic matters so that nutrients will become available for the growth of the natural fish food. Lastly, is to kill disease, which is causing microorganisms. Next, we have the tilling or cultivating the pond bottom. So this is done with the use of either rotavator, plow, or rake for a small pound. The purpose of this is to make the subsurface nutrients available at the surface for the growth of natural fish food, to eradicate the burrowing fish enemies, and to eliminate and destroy the pond weeds. So tilling will expose subsurface nutrients, control weeds, and eliminate burrowing pests, improving pond productivity. So after tilling, we will level the pond bottom. So leveling will ensure that proper drainage and prevents the water stagnation, which is important for pond health and management efficiency. Next is we have the soil sampling. So the purpose of soil sampling is to assess the physico-chemical condition of pond soil, to check organic matter, nutrients, pH, and other properties. It also helps in determining the proper soil conditioning and treatment before fish culture. Then, how do we take the sample of the soil? or the soil. So we will collect samples from each pond compartment by following the S-shaped sampling pattern and must include the both surface and the subsurface soil. For sample preparation and analysis, some samples will follow this step. So air dry under shade, then homogenize, then afternoon is yung nutrient and organic matter analysis. Then the organic matter will be determined using oven method. Other samples will be tested immediately for pH.
For this part, I will be discussing about the soil conditioning. So soil conditioning is the process of improving soil from an undesirable or poor condition to a normal and suitable state. This is done by adding neutralizing or buffering materials such as lime or acid-forming substances like alum or gypsum depending on the soil or water pH. So when the soil or water is acidic, lime should be applied to raise the pH and make the environment more suitable. And on the other hand, when the soil or water is excessively alkaline or has a very high pH, acid forming fertilizers like alum or gypsum are used to lower the pH and improve the soil condition. Alum or aluminum sulfate works by hydrolyzing in water and releasing hydrogen ions, which help reduce the alkalinity. Meanwhile, gypsum or the calcium sulfate increases calcium hardness in the soil or water, which leads to the precipitation of calcium carbonate that helps stabilize pH and improve soil quality. In general, it is recommended to maintain total alkalinity and total hardness within the range of 20 to 200 milligrams per liter as calcium carbonate to ensure optimal soil and water conditions. Now, let's move on to liming. Liming refers to the application of lime to pond soil or water in order to improve its quality and make it suitable for aquatic production. First, liming helps neutralize acidity or sourness in the soil or water as it buffers the pH and brings it to an acceptable alkaline level that is favorable for aquatic organisms. Second, liming can help kill parasites, pests, and other undesirable organisms present in the pond, making the environment safer for culture species. Third, it helps improve soil condition and promotes the activity of the beneficial bacteria. These bacteria break down waste materials and organic matter, which improves pond productivity. Fourth, liming aids in the precipitation of excess dissolved organic materials and clay particles, reducing the turbidity and making the water clearer. Lastly, liming can help reduce the risk of oxygen depletion by making carbon dioxide available for photosynthesis, which supports the production of oxygen by aquatic plants and phytoplankton. The common materials used for liming includes limestones, oyster shells, and other basic substances. These materials typically contain compounds such as calcium carbonate or magnesium carbonate as well as calcium hydroxide and similar alkaline substances that help raise the pH and improve pond condition. Next is lime. So what is lime? Lime is a material derived from limestones and shells such as oyster shells and it mainly contains calcium carbonate. Lime is commonly used in ponds to treat or condition the soil and water by improving pH and overall quality. There are several kinds of lime used in pond management. First is the pulverized limestone, also called as agricultural lime, which contains calcium carbonate. Second, burnt limestone, known as quick lime or unslaked lime. Third, hydrated lime, also called as slaked lime. And fourth, dolomite lime, which contains both calcium carbonate and magnesium carbonate. Now, let's start with the agricultural lime and the dolomite lime. So they are produced by crushing limestone into small particles using a rock crusher. These particles usually range from 10 mesh, which is about 1.70 millimeters in diameter, down to 60 mesh, which is very fine at less than 0.24 millimeters in diameter. The finer the particle size, the faster the lime dissolves. In fact, the particles that pass through a 60 mesh screen have finest value of 100%, meaning they dissolve the fastest and react more quickly in the pond soil or water. Agricultural lime has a neutralizing value equivalent to 100% calcium carbonate. Although it is relatively slow acting, it is widely used because it is low in cost and safe for pond application. This type of lime is commonly used or mainly used to neutralize acidity and increase pH in pond soil and water. It also helps in the precipitation of suspended clay and silt particles as well as excess dissolved organic matter, which improves water clarity and overall pond condition. Next is the burnt limestone, also known as quicklime, and is produced by heating limestone in a furnace at very high temperatures, usually around 500 to 600 degrees Celsius. During this process, calcium carbonate is converted into calcium oxide with carbon dioxide released as a gas. Burnt limestone has a neutralizing value of about 173 to 179% compared to calcium carbonate, which makes it very strong and effective. Because of this, it is considered the fastest acting type of lime used in pond treatment. It is commonly applied to neutralize acidity and rapidly increase pH, and it also helps in the precipitation of excess dissolved organic matter and suspended clay or silt that improves pond condition and can be used to kill pests, parasites, and disease-causing organisms. However, burnt limestone is highly caustic. It can irritate or damage the skin and eyes if handled improperly. Therefore, proper care, protective equipment, and caution must always be observed when using this type of lime. And lastly, the hydrated lime, also known as the slake lime. It is produced by adding water to burnt lime or quick lime. In this reaction, calcium oxide combines with water to form calcium hydroxide. Hydrated lime has a neutralizing value of about 135 to 136% compared to calcium carbonate, making it a strong liming material. It is also fast acting, although it works slightly lower than burnt lime. This type of lime is used to neutralize acidity and raise the pH of pond soil and water. It also helps in the precipitation of excess dissolved organic materials and suspended clay or silt particles, improving pond condition and can be applied to kill pests, parasite, and disease-causing organism. However, like burnt lime, hydrated lime is caustic and can irritate or damage the skin and eyes, so proper handling, protective equipment, and caution are necessary when using it. How do we distinguish different types of lime? So to distinguish one type of lime from another, we can examine the pH of a slurry made by mixing the liming materials with distilled water. So the usual mixture is about 10 to 20 parts distilled water to 1 part lime, or a 10 to 20 to 1 ratio. So after mixing, the pH of the slurry is tested. So if the material is agricultural lime or dolomite lime, the pH of the mixture will usually not exceed with a pH of 10 because these types are milder and slower acting. However, if the material is burnt lime or hydrated lime, the pH of the slurry will exceed with a pH of 12. So this is because these forms of lime are stronger, more reactive, and more alkaline. So by checking the pH level of the slurry, we can easily determine whether the lime is a mild agricultural lime or a stronger, fast-acting lime such as burnt or hydrated lime. There are some practical ways to determine if pond soil is acidic and needs to be neutralized by liming. So first is by through ocular inspection. So the indication of acidic pond soil are as follows. First, a newly constructed pond is likely to be acidic because the soil has not yet been stabilized. Second, ponds that do not respond to fertilization may indicate acidity since nutrients are not easily utilized under low pH condition. Third, ponds with plenty of decaying bakawan and nipa roots are often acidic because decomposing plant materials release acids into the soil. Fourth, when the pond bottom turns reddish after being exposed to sunlight for more than 3 days, this is a sign of acidic soil due to oxidation. Fifth, the presence of jarosite, which can be identified by a yellow coloration of the soil, is another clear indicator of acidity. Lastly, if the pond water has alkalinity of less than 20 ppm or a total hardness of less than 20 milligrams per liter as calcium carbonate, the soil and water are likely acidic and require liming. Second, or the another practical way to determine acidity is by using litmus paper. If litmus paper turns reddish after being moistened with the pond water, this indicates that the water is acidic. If the litmus paper turns bluish, the water is alkaline. Acidity can also be determined using a chemical indicator mixture. So the mixture commonly used for pond soil and water testing contains zero point 0.8 grams of bromothymol blue indicator, zero point 0.4 grams of methyl red powder, and zero point 0.2 grams of methyl orange powder. So these three chemicals are dissolved together in 1 liter of distilled water to prepare the indicator solution. After treating pond soil with a chemical indicator mixture, the color that appears can be used to determine the soil condition and estimate its pH. If the soil turns reddish, it is considered very acidic with a pH of 4 or lower, and if the soil turns yellowish, it is slightly acidic with a pH between 5 and 6, And if the soil turns greenish or grayish, it is neutral with a pH range of 6.5 to 8.5. By observing these colors, pond managers can decide whether liming is necessary to adjust soil acidity and improve water and soil condition for aquatic life. So let's discuss about the major sources of acidity in pond soil. First, vegetation that produces tannic acid, such as rhizophorus species, commonly known as bakawan, can make the soil acidic as these plant materials decompose. Second, soil sulfides contribute to acidity. When soil sulfides oxidize, they produce sulfuric acid, which lowers the pH of the pond soil and water. Third, runoff from upland areas or pond dikes can carry acidic elements into the pond, further increasing acidity. So there are two main methods to control and neutralize acidity. First is washing or flashing, also called as leaching. So this involves draining and drying the pond so the soil is exposed to air and sunlight. Afterward, the pond is filled with water and washed several times to remove acids and improve soil condition. Second is liming, which involves applying lime to neutralize acidity and raise the pH to a suitable level of aquatic organism. So together, these two methods help maintain pond soil and water in a healthy, productive condition. In liming ponds, proper application of lime is essential to effectively neutralize soil acidity, improve soil quality, and prepare the pond for fertilization and production. So correct procedures must be followed to ensure that liming achieves maximum efficiency. So first, the pond soil should be analyzed to determine whether it is acidic or alkaline and to assess its nutrient or organic matter content. This step helps identify the appropriate type and amount of lime needed. Second, once the need for liming is confirmed, the lime should be broadcast or evenly spread over the entire pond bottom, and the pond must be drained but still moist since moisture allows better reaction between the lime and the soil. Third, the applied lime must be thoroughly mixed with the soil, and proper mixing ensures uniform distribution and allows the lime to react efficiently, so resulting in better neutralization of acidity. So to determine the amount of lime required, the following formula is used. So where QL is the quantity of lime required, is the desired pH, APH is the actual average pH of the pond soil, and VL is the neutralizing value of lime in decimal form, and A is the pond area in hectares. And now let's go through an example of calculating the quantity of lime required for a pond with a desired pH of 6.8 and an actual pH of 6.5 in a 1 hectare pond. So we use the formula of quantity of lime required equals to the desired pH minus the actual pH times zero point 0.5 ton per hectare times the area in hectares over the zero point 0.1, which is the standard pH correction unit, and the neutralizing value of lime. So for each type of lime, there is a different neutralizing value or NVL because each material differs in its chemical strength in neutralizing acidity. Agricultural lime has an NVL of 1.0, quick lime has an NVL of 1.73, and hydrated lime has an NVL of 1.35. Once we already have the desired pH, the actual pH, and the area of the pond, we substitute those values into the formula. After substituting, the only value that changes is the NVL depending on which type of lime we are using. So to determine the required amount of lime, we simply insert the appropriate NVL into the formula and solve. The higher the NVL, the stronger the lime and the smaller the quantity needed. For this slide, this table shows the recommended lime application rates per hectare based on soil pH. The more acidic the soil, the more lime is required. For pH below 5, the highest amounts are needed. As pH increases from 5 to 6 and 6 to 7, the required lime decreases. So among the three types, quick lime requires the smallest amount because it has the highest neutralizing value, followed by hydrated lime, while agricultural lime requires the largest amount. In summary, lime requirement decreases as soil pH increases, and the higher neutralizing value means lower application rate. Before applying lime in ponds, there are several important considerations that must be taken into account to ensure proper and effective liming. Not all pond soils require lime treatment. For instance, sandy soils with a pH ranging from 6.1 to 6.5 are already near neutral and may not need to be treated with lime. Because of this, the decision on what type of lime to use must be carefully evaluated based on the soil condition and management objectives. Liming should not only be used to neutralize acidity but also enhance the decomposition of an organic matter in the pond bottom. However, the use of quicklime or hydrated lime, which have a very high calcium carbonate efficiency, may kill beneficial microorganisms. When this happens, the decomposition of organic matter may be temporarily stopped or slowed down. Therefore, agricultural lime is generally preferred when the goal is to support microbial activity and improve organic matter breakdown. Lime should be applied on the pond bottom surface as well as on the top and side of the pond dikes to neutralize acidity and possibly eliminate disease-causing organism. After proper liming has been completed, the pond becomes ready for fertilization and further preparation for culture operations.
Next topic for chapter 4 are fertilization and fertilizers. So fertilization, it refers to the addition of organic and or inorganic fertilizers to soil or water to stimulate and maintain growth of desirable phytoplanktons and other kinds of algae for food of animal organisms including fish. On the other hand, fertilizers refers to organic and or inorganic fertilizers. So there are two general classes of fertilizers, the organic fertilizers and the inorganic fertilizers. Organic fertilizers are fertilizers capable to be decomposed and which contain varying amounts of nutrients. On the other hand, inorganic fertilizers are fertilizers that are not decomposed but rather dissolved in water and which contain concentrated amount of at least one of the three major plant nutrients, the NPK, or also known as nitrogen, phosphorus, and potassium. Like what I've said, organic fertilizers are fertilizers capable to be decomposed and which contain varying amount of nutrients. So examples, first is animal manure, dung or droppings from hog, cattle, poultry, and birds. Another examples are green manures from leaves of legumes, grasses, rice straw, and rice bran. Lastly, the compost plant and animal materials. Below is a table of the average composition and percentage of several manures. On the other hand, inorganic fertilizers. So these are fertilizers that are not decomposed but rather dissolved in water and which contain concentrated amount of at least one of the three major plant nutrients, the NPK or nitrogen, phosphorus, potassium. So there are three classification of types of inorganic fertilizers. The single element fertilizers, the incomplete fertilizers, and lastly the complete fertilizers. So, for the classification or types of inorganic fertilizers, first we have the single element fertilizers, are those which contain only one fertilizer element. So, below is a table of example of single element fertilizers. So, for nitrogenous fertilizers, we have here urea and ammonium sulfate, which has 46 and 21 nitrogen respectively with no amount of phosphorus and potassium. Another one are phosphatic fertilizers, and we have here the superphosphate and cellulose, which has 20 and 18 phosphorus respectively, with no amount of nitrogen and potassium. And lastly, the potassic fertilizers. We have here the murate of potash and potassium sulfate, which has 60 and 48 potassium respectively, with no amount of nitrogen and phosphorus. Another classification or type of inorganic fertilizers are the incomplete fertilizers. So, are those which contain two fertilizer elements. So, we have an example here, ammonium phosphate, which has 16 nitrogen, 20 phosphorus, but no amount of potassium. Another one is the diammonium phosphate, which has 18 nitrogen, 46 phosphorus, but no amount of potassium. Another classification or type of inorganic fertilizers are complete fertilizers. So, are those which contain all three major nutrients. Determination of amounts of fertilizer nutrients and of the required absolute amounts of fertilizer. First is how to determine available amounts of nutrients, the NPK, present in a given inorganic fertilizer. So for that, the formula is weight of nutrients available is equals to the weight of fertilizers multiplied the percentage of fertilizer nutrient available for use in decimal. So first example here is 50 kilogram urea, which has a nutrient ratio of 46% nitrogen, 0% phosphorus, and 0% potassium. So first is step 1, or the step A is to calculate the nitrogen content, which is 0.46 multiplied to the 50 kilogram is equals to 23 kilogram nitrogen. So 23 kilogram, we subtract the 23 kilogram nitrogen to the total weight of the fertilizer, 50 kilogram, which gives us a 27 kilogram filler. Therefore, out of 50 kilogram urea, 23 kilogram is nitrogen and 27 kilogram is filler material. So, to further understand, another example is given. So, number two example is 50 kilogram ammonium phosphate, which has a nutrient ratio of 16% nitrogen, 20% phosphorus, and 0% potassium. So, step 1 or step A is to calculate the nitrogen content. So, 0.16 multiplied to 50 kilogram is equals to 8 kilogram nitrogen. So, step 2 or step B is to calculate the phosphorus content. 0.20 times 50 kilogram is equals to 10 kilogram phosphorus. Then step C or step 3 is the potassium content, which is 0 times 50 kilogram is 0 kilogram potassium. So, add the total nutrients available, 8 kilogram nitrogen plus 10 kilogram phosphorus is equals to 18 kilogram of nutrients. So, subtract this 18 kilogram nutrients from the 50 kilogram of total weight of the fertilizer, which is 50 kilogram, which gives us 32 kilogram filler. So, therefore, out of 50 kilogram ammonium phosphate, 8 kilogram is nitrogen, 10 kilogram is phosphorus, and 32 kilogram is a filler material. How to determine the required amounts of inorganic fertilizers with the given recommended or required treatment or dosage. So there are two options as a formula. Option 1 is the amounts of fertilizer in kilogram. So it is equals to the recommended treatment or dosage of nutrients in kilogram divided by the percentage of fertilizer nutrients available for use in decimal. Another option is the amount of fertilizer in bag is equals to the dosage of nutrients in kilogram times 100 divided by the weight of fertilizer per bag multiplied to the percentage of fertilizer nutrient. So, to further understand, first example here we have the recommended dosage of a nitrogen, 40 kilogram, phosphorus, 0 kilogram, and potassium, 0 kilogram. To solve, so for option 1 formula, in amounts of fertilizer in kilogram, so 40 kilogram nitrogen divided by the 0.46 nitrogen and urea. So it's equals to 86.96 kilogram urea. So for option 2 formula, which is in the amount of fertilizers in bag, so it's 40 kilogram nitrogen times 100 over to 50 kilogram per bag times 46% nitrogen and urea. So it's 4,000 divided by 2,300. So it's equals to 1.74 bags of urea or 86.96 kilogram. So therefore, to apply 40 kilogram of nitrogen, use 86.96 kilogram of urea or 1.74 bags.
Now, how to determine amount of fertilizer to be applied using a ratio and proportion formula or cross multiplication technique. So this is the formula shown here. A is to B is equal to C is to D, or A over B cross multiplied into C over D. So an example of problem solving. The standard amount of or rate of fertilizer application is 50 kilograms per hectare of ammonium phosphate. How much amount of the same fertilizer is needed if the area to be fertilized is only 3,500 square meters? So the first thing that we should do is to write down the given information. So the standard fertilizer rate is 50 kilograms per hectare of the ammonium phosphate fertilizer. Next, the next given detail is area to be fertilized, which is 3,500 square meters. Then we must know that the conversion of 1 hectare is equal to 10,000 square meters. So to solve the problem, there are three options that we can follow, which is the first one, the most direct, is to convert area to hectares first. So 3,500 square meters divided into 10,000 square meters is this gives us 0.35 hectares. Then the next step is to apply the fertilizer rate. So we multiply the rate, which is 50 kilograms per hectare, by the actual area in hectares. So that is 50 times 0.35. The result is 7.5 kilograms. Therefore, the required fertilizer is 17.5 kilograms of ammonium phosphate. The next method is ratio and proportion, or using the cross multiplication. So the rate 50 kilograms is to 1 hectare is equal to x is to 0.35 hectares. We will cross multiply. This will give us x is to 50 times 0.35 over 1. Then the result will be the same, 17.5 kilograms. The third method is to convert rate to kilograms per square meters. This is the opposite of the first method, wherein instead of converting the square meters into hectares, we will convert the hectares into square meters. So 50 kilograms over 10,000 square meters is equal to 0.005 kilograms per square meters. Then we will multiply the area, 0.005 kilograms per square meters times 3,500 square meters, the given, which will result to 17.5 kilograms. So all the three methods agree. Then the required fertilizer is 17.5 kilograms of ammonium phosphate. Next, how to determine the amounts of fertilizer needed to apply when the treatment or dosage is in parts per million. So 5 parts per million, which means 5 milligrams per liter or 5 grams per cubic meter of water. The problem is how much amount of ammonium phosphate will be needed in a 1 hectare pond having a water depth of 1 meter and requiring a treatment or dosage of 5 ppm. The formula is the amount of fertilizer equals volume of water times dosage of fertilizer in parts per million. So first, we must note the given, which is the pond area is equal to 1 hectare or 10,000 square meters. Then the water depth is 1 meter. The target dosage is 5 parts per million.
And in conversion, for water, 1 ppm will equate to 1 milligram per liter or 1 gram per cubic meter. To solve, we must first compute the water volume. Volume equals area times depth. 1 hectare is 10,000 square meters multiplied by 1 meter depth. So the volume is 10,000 cubic meters. Then the next step is to convert the ppm to usable units. In water, 1 parts per million equals 1 gram per cubic meter. So 5 ppm equals 5 grams per cubic meter. Then the third step is to compute the fertilizer needed. Multiply the volume by the dose, that is 10,000 cubic meters times 5 grams per cubic meter. This equals 50,000 grams. Then the last step is to convert the grams to kilograms. So divide by 1,000, that gives us 50 kilograms. Therefore, the required fertilizer is 50 kilograms. Next, to find the dosage of fertilizer in parts per million when the amount of fertilizer to be applied and the volume of water are given is to use the following formula. Dosage of application in parts per million equals amount of fertilizer to be applied in grams over actual volume of water. So example, the amount of fertilizer to be applied to the whole area is 50 kilograms or 50,000 grams per hectare. Then the volume of water in a hectare of pond is 10,000 cubic meter. So to solve, fill in the data into the formula. 50,000 grams over the 10,000 cubic meter is equal to 5 grams per cubic meter or 5 parts per million.
Moving on to how to determine the equivalence of amount of inorganic fertilizer to animal manure basing on one of the NPK content. Say na hinahanap lang po natin is yung P or the N of the fertilizer. So for the example for the above problem, if 1 hectare pound would require 50 kilogram of inorganic fertilizer having a phosphorus content of 20%, how many kilograms of organic fertilizer containing 0.41% phosphorus would be needed as equivalent of the former fertilizer in terms of phosphorus content. So for the solution, the first step is that we need to determine the amount of phosphorus in kilogram in the 50 kilogram inorganic fertilizer like 16, 20, and 0. Thus, the 15 kilogram should be multiplied by 0.2 P kasi 16, 20, 20 yung P. Then, ang magiging answer is that 10 kilograms P. So for the second step, we need to determine the amount of the animal manure in kilogram using an algebraic solution. So we take the 0.41% phosphorus na nandoon sa problem and we convert it into decimal. So 0.0041 px equals 10 kilogram P in 1620 zero fertilizer. So to find x, we need to divide 10 kilogram P by 0.0041 P. So the P will be cancelled. May iwan na lang yung kilogram and the answer is 2,439 kilograms or 2,400 kilograms animal manure is needed. So to check the equivalent amount of phosphorus in animal manure is to do the following. So we take the kilogram of manure, the 2,439, and multiply it by 0.0041 P. So the answer is 10 kilogram P in manure. Lastly is how to determine the exact rate of organic fertilizer application with the consideration of the following. First is the organic matter of the soil, second is the organic matter of manure, and lastly the depth of surface soil that benefits from fertilization. Shown here is the formula. AM is equals to B multiplied by C minus X over X minus D, where AM is the amount of animal manure, B is the weight of 2 centimeter or 0.02 meter depth of surface soil in an area to be fertilized. C is the original organic matter content of the subject pond soil. And D is the organic matter content of the poultry manure. And X is the desired organic matter content of the subject pond soil. For example, given the following that the B is 200 tons, C is 7%, D is 40% organic matter content of poultry manure, and X is 8%. So to compute for the AM, we need to substitute the given values in the formula. So it is shown here, the substitution, 200 tons multiplied by 7% minus 8% over 8% minus 40%. So, ginawa yung percentage into decimal point and the operations are applied. So, the answer is 6.25 tons per hectares. Moving on to the common practice on rate of fertilization application. So the recommended rates of chicken manure application are based on the pond condition and soil type when no soil analysis is available. So for a newly constructed ponds with sandy loam soil, a higher rate which is 3,000 to 4,000 kilograms per hectare combined with 50 kilograms per hectare urea is applied as the basal fertilization. It is to enhance mineralization and also stimulate initial plankton production. For older ponds naman with loamy and clayey soils, a lower rate of 1,000 to 2,000 kilograms per hectare with 50 kilograms per hectare urea is sufficient because these ponds already contain more accumulated organic matter. Lastly, the 1,000 to 2,000 kilograms per hectare rate may also be applied every 2 weeks, stopping 30 days before harvest to maintain plankton, which supports fish growth. So overall, the purpose of these application rates is to ensure that adequate natural food production in the pond for better fish productivity. For inorganic fertilization naman, when no soil analysis is available, a basal application of 50 kilograms per hectare of complete fertilizer is recommended. Use 16-20-0 for brackish salty water ponds and 14-14-14 for freshwater ponds. So this provides essential nutrients, particularly nitrogen and phosphorus, which stimulates the plankton growth. To maintain the plankton production, 8 kilogram nitrogen and 10 kilogram phosphorus per hectare should be applied every 2 weeks, stopping 2 weeks before harvest. This rate is considered optimal for sustaining the growth of lablab, which serves as natural fish food. For the proper application of fertilizer, dissolve one-third to one-half of the total fertilizer in a pail of water and broadcast the solution evenly across the pond surface for immediate nutrient availability. The remaining fertilizer is wrapped in a cloth and either hung on stakes or placed on a platform in the pond to allow this slow and gradual dissolution, ensuring that there is a continuous nutrient supply in the pond.
For inorganic fertilization in fish ponds, it's important to pay attention to timing and weather conditions. Fertilizers should not be applied during heavy cloudy or rainy days because phytoplanktons and other beneficial algae do not respond well in these conditions. The best time to apply is on a sunny day, specifically between 9 a.m. and 12 noon, to ensure the nutrients are quickly assimilated by plant organisms. Additionally, water should only be changed every 13 to 14 days after fertilization to prevent the early loss of nutrients. Following these practices help maintain optimal water quality and encourages healthy growth of algae that supports fish development. For organic fertilization, caution is also necessary, especially after stocking the fish. Organic fertilizers should not be immediately spread across the entire pond surface because this can disrupt water quality and harm the fish. Instead, the fertilizers should be soaked in a container or bag first, and then released gradually into specific areas of the pond. This method ensures that nutrients are absorbed properly without causing sudden changes in water chemistry, which can negatively affect fish health and growth. When propagating lablab in a fish pond, fertilization begins with applying chicken manure evenly across the pond bottom. The recommended rate is 1,000 to 2,000 kg per hectare for new ponds that are 1 to 4 years in use, and 500 to 1,500 kilograms per hectare for older ponds. After spreading the manure, water is admitted to a shallow depth of about 3 to 5 centimeters, followed by the application of 500 kilograms per hectare of urea to accelerate manure decomposition. The pond should be allowed to dry for 3 days before water is readmitted to a slightly deeper level of 8 to 10 centimeters. These steps help prepare the pond environment and nutrients for lablab growth. The next step involves adding mineral fertilizers to support lablab growth. On the following day, 16-20-0 fertilizer at 100 kilograms per hectare or 18-46-0 at 50 kilograms per hectare is spread evenly across the fish pond. Water depth is gradually increased by 5 centimeters every 3 days until it reaches 20 centimeters. During the rearing period, 16-20-0 fertilizer is applied at 15 kilograms per hectare every 7 days, but application must stop at least 3 days before fish stocking. These practices ensure a steady supply of nutrients for the lablab while maintaining water quality suitable for fish. 3 days before stocking, about 25% of the pond water is gradually drained and refilled to reach the desired depth. 20 to 30 centimeters for NP ponds, 30 to 40 centimeters for TP ponds, and 50 to 60 centimeters for RP ponds. Fish are typically stocked 28 to 35 days after manure application, depending on the thickness of the lablab, which should be around 1 centimeter. To maintain a luxuriant growth of lablab, a side dressing of 16-20-0 fertilizer at 15 kilograms per hectare is applied every 2 weeks. But all fertilization should stop 20 days before harvest. This method ensures that the lablab provides optimal coverage and nutrients for fish while promoting sustainable pond management.
Now let's proceed to the production of plankton. First, fill the pond with water to a depth of at least 60 centimeters, preferably 75 to 100 centimeters. Apply 1 to 2 tons per hectare of chicken manure contained in bags by soaking them in the pond for at least 24 hours before releasing. Add 50 kilograms per hectare urea after the manure had settled down, and organic fertilization along with urea, which serves to hasten decomposition of manure, could also be done before addition of water into the pond. Then, apply ammonium phosphate at 50 kilograms per hectare or diammonium phosphate, which is 22 kilograms per hectare, 1 week after the first fertilizer application. One half of this amount will be dissolved in pail of water and spread over the pond water surface. The other half will be put in a thin cloth bag or any substitute and then hang strategically on a stake positioned in the pond for gradual release of fertilizer nutrients. Then observe productivity of plankton for the next 7 days through the green to yellowish green turbidity of water. Visibility reading using a device called Secchi disc is ideal at 30 to 40 centimeters below water surface. Turbidity above this range indicates low plankton productivity and signals further or additional fertilization. Then fish stocking shall be done 2 weeks or so after filling of water, during which time plankton growth is at 30 to 40 centimeters Celsius level. Lastly, apply 50 kilograms per hectare of phosphate or 20 kilograms per hectare of diammonium phosphate every 2 weeks plus 1,000 kilograms per hectare chicken manure every month thereafter. Then to maintain the abundance of plankton. Stop fertilization 20 days before harvest. Then cultivation of lumot or the filamentous algae. They grow favorably in lower salinities, less than 25 parts per thousand, and a water depth of 20 to 60 centimeters. In growing, the same fertilization program used for lablab production is applied. Lumot occurs naturally, but seeding may be resorted to when growth is sparse. Fibrous or silky filaments are best type of life stage of lumot for seeding or planting purposes. So these are planted either by staking, sowing, or spreading. Luxuriant growth is not entirely desirable because this plant and the fish stock will compete for living space.